Hello class, let's continue to study Chapter 3. We have learned about the structure of cell walls. In this lecture, we are going to learn about the bacterial cell membrane structure in detail. The cell membrane, also named as cytoplasmic membrane, plasma membrane, inner membrane. It is a thin structure lying inside the cell wall and enclosing the cytoplasm of the cell. As indicated by red arrow in electron micrographs, prokaryotic and eukaryotic plasma membrane look like two-layered structures. There are two dark lines with a light space between the lines. For example, as shown in the figure, this is a micrograph of photosynthetic membrane stacks derived from the cytoplasmic membrane in the phototrophic bacteria. Its bilayer is about 8 nanometers thick. The dark lines between light lines in micrograph is a region of fatty acids. The light lines in micrograph is a glycerol portion of phospholipid. The phospholipid molecules are arranged in two parallel rows, called a lipid bilayer. Each phospholipid molecule contains a polar hand, composed of a phosphate group and glycerol that is hydrophilic and soluble in water, and nonpolar tails, composed of fatty acids that are hydrophobic and insoluble in water. The polar hands are on the two surfaces of the lipid bilayer, and the nonpolar tails are in the interior of the bilayer. Two types of membrane proteins have been identified based on their ability to be separated from membrane. Peripheral membrane proteins are loosely connected to membrane and can be easily removed. They are soluble in aqueous solutions and make up about 20 to 30% of total membrane protein. The remaining proteins are integral membrane proteins. These are not easily extracted from membranes and are insoluble in aqueous solutions when freed of lipids. Integral membrane proteins, like membrane lipids, are amphipansic. Then hydrophobic regions are buried in the lipid, while the hydrophilic portions project from membrane surface. Integral membrane proteins carry out some of the most important functions of the membrane. Many are transport proteins used to move materials either into or out of the cell. Others are involved in energy-conserving processes. Those integral membrane proteins with regions exposed to the outside of the cell enable the cell to interact with its environment. Studies have demonstrated that the phospholipid and protein molecules in membranes are not static but move quite freely within the membrane surface. This movement is most probably associated with many functions performed by the plasma membrane because the fatty acid tails cling together. Phospholipids in the presence of water form a self-sealing bilayer. As a result, breaks and tears in the membrane heal themselves. The membrane must be about as viscous as olive oil, which allows membrane proteins to move freely enough to perform their functions without destroying the structure of the membrane. This dynamic arrangement of phospholipids and proteins is referred to as the fluid mosaic model. Bacterial membranes usually differ from eukaryotic membranes in lacking steroids such as cholesterol. However, many bacterial membranes contain steroid-like molecules called hoponoids. Hoponoids are synthesized from the same precursors as steroids, and like the steroids in eukaryotic membranes, they probably stabilize the membrane. Because bacteria have none of the eukaryotic organelles, the cell membrane provides a set for energy, nutrient processing, and synthesis. A major action of the cell membrane is to regulate transport, that is, the passage of nutrients into the cell and the discharge of waste. The cell membrane is also involved in secretion or release of metabolic products into the extracellular environment. The membranes of bacteria are an important site for a number of metabolic activities. Okay, that's all for today's lesson.